So I have the pleasure of introducing the next speaker, who is Tyler Cassidy. He's an applied mathematician working at the intersection of medicine, and he's currently doing his postdoctoral research in the theoretical biology group at Los Alamos National Laboratory. His research has developed a whole bunch of models, including structural population models and delay differential equation models that have been really helpful for understanding cancer cell cycling and the effects of treatment. And his work has also involved PK/PD modeling and virtual clinical trials. So I'm really looking forward to this talk, hearing all about your models, Tyler, and I'll let you take it away. Thank you.
Perfect, thanks. Yeah, it's a pleasure to be here, and it's been a really enjoyable 3 days for me to listen into some of the other research that's going on. So it's nice to get to contribute a little bit at least. And maybe the two themes that I've really taken away from the workshop so far have been kind of two contrasting ways of looking at heterogeneity. So we've talked, a lot about these virtual clinical trials and how we can try to understand entire populations with, the combination oncolytic virus work that was done, that was spoken about on the first day, or even some of Richard's work earlier today. And then with the agent-based modeling that we talked about yesterday, we talked really about this intra-patient heterogeneity. So this is heterogeneity inside a certain patient, and this might be, this might come into different cell types or how different genes are represented on genetically identical cells or what have you. And the idea for today is, for my talk at least, is to try to bridge this gap. And really the goal here is to show one perspective of how we can try to incorporate some intra-patient heterogeneity. So this is, heterogeneity inside a single patient into the framework we have to understand inter-patient heterogeneity, or trying to include this intra-patient heterogeneity inside a virtual clinical trial. And to do this, I really have two kind of basic goals. One is that, this modeling should be biologically motivated. So we should start from some sort of real heterogeneity that matters. And then secondly, it has to be computationally efficient. And what I mean by this is, we don't want to just have our arbitrary threshold of 100 patients in our virtual clinical trial because our model is too difficult to run. Rather, we want to be able to come up with a model that both includes in-patient heterogeneity in a way that's biologically motivated, but is also, somewhat simple to run, thousands, 10,000s, or hundreds of thousands of times that we're going to need for a virtual clinical trial. And the framework I'm going to present here fits maybe a bigger set of heterogeneity than what I'm going to present, but I'm really going to focus on kind of a worked example about the cancer cell cycle. So the data I'm showing here is from a paper in 2016 that basically took colorectal cancer cells, and they would follow these cells in addition. They would start a timer when a mother cell would divide, and then they would stop that timer when the two daughter cells then reproduced again. So essentially, the timer was modeling or keeping track of how long, what the intermitotic time is, or how long it takes for a population to double, or a single cell to go from one cell to two cells to four cells. And what they've plotted here is essentially a histogram of their results. And these cells are genetically identical. They're living in the same dish. They should have access to the same resources. And what we see is that some cells will divide very, very quickly, kind of at this 20-hour range. And then there's some sort of distribution of intermitotic times or doubling times that goes from 20 cells all the way up to 60 cells. And for some reason, the authors decided to fit a normal distribution to this data. I don't want to talk about that decision, but rather I'm more interested in the fact that there is this really heterogeneity in a genetically identical population of cells in a dish in terms of how long it takes them to divide. And the statistic up here is showing that on average, it takes these cells about 33 hours to complete one division, and this is plus or minus 1 standard deviation, which is about 7 hours. And this data is going to be important a little bit later. So why might there be this heterogeneity? Well, if we really go back to what is happening inside a cell that's actively dividing, or a cell that's inside the cell cycle, there's a number of different processes that have to occur. So we can have cells that are actively dividing. These cells might be in, say what we would call GAP1 or G1. Cells in G1 are essentially growing to get ready for division and mitosis. The second stage of the active portion of the cell cycle is essentially what's called the S phase, which corresponds to DNA synthesis, so essentially creating 2 copies of DNA. There's a second growth phase, GAP2, before finally progressing into mitosis and actually completing division. And the duration of this process is, like we showed on the previous slide, going to be variable between cells. And I want to somehow model this. And to model this, I'm going to consider two distinct populations. I'm going to consider a population of resting cells. So these are cells that aren't actively dividing. And throughout the rest of the talk, we can think about, I'm going to denote them as x of t. And then the second population where all the action is happening are these actively dividing cells. And here, I'm going to use n of t comma xi, where xi essentially represents some level of heterogeneity for a population of cells. So this xi could be how old the cell is, how large the cell is, what stage of the cell cycle the cell is in, what genes are being expressed, what the phenotype is, so on and so forth. So this xi variable essentially is going to encode the intra-patient heterogeneity that I mentioned at the very beginning. And because I'm going through a worked example, I don't want to think too hard about this. I'm just going to think about xi representing the age of the cell, or how far that cell is into the active portion of the cell cycle. And that's a little bit weird to think about in this circle diagram. So let's take the active portion of the cell cycle, so G1, S, G2, and then into mitosis, and unravel this circle. Oh, pardon me, I'm one slide ahead of myself. What I meant to say is that the portion of the dynamics of the resting cells or G0 cells, we can essentially write as just a normal differential equation, which depends, on the number of G0 cells right now, but then also on the number of cells that are exiting the cell cycle, or what I'm going to call the mitotic output. And I'm going to say that these G0 cells will enter into the active portion of the cell cycle at some rate beta. And this entrance into the active portion of the cell cycle gives me a boundary condition, or it gives me the density of cells that are of actively dividing cells with some structure variable xi0. And when we're talking about age, we can just set xi0 to 0, because cells that are entering into the active portion of the cell cycle have, kind of a 0 age for this portion. Now we get to unravel this circle of the active portion of the cell cycle, and we can just kind of think about it as a conveyor belt. So cells will get placed on this conveyor belt when they enter into the first stage of the active portion of the cell cycle, or G1, and then they're going to move along this conveyor belt going through G1, through S, through G2, and then finally into mitosis when they fall off the end of the conveyor belt and enter back into the quiescent population. And if we take this idea of having a conveyor belt and write it down in a mathematical formalism, we end up with this partial differential equation on the bottom here. So the left-hand side, which is essentially the time derivative and the age derivative, is representing how a population of cells or a density of cells moves along the conveyor belt. And the right-hand side is showing what happens to this population of cells as they progress through the cell cycle, or as they progress along the conveyor belt. And we're going to have essentially two loss terms. One is going to be this gamma of t comma x of t. This loss term can represent essentially anything we want to throw at the model. We could think about this as being apoptosis, which is just random cell death. We could think about this as being immune predation. We could think about it as being treatment. We can really throw whatever we want inside this gamma term. It doesn't really bother us. And the second loss term is going to be this term h of t comma a that I've highlighted in red. These are cells that are leaving the active portion of the cell cycle because they've actually divided. They've finished the active portion of the cell cycle. And what this corresponds to is not having a conveyor belt like I've shown here with a very well-defined edge, because if that would give us no heterogeneity in the cell cycle, but rather some kind of probabilistic way of understanding when cells are likely to finish the cell cycle. And just as an example of what types of functions we can use for gamma, or where this kind of formalism might show up in cancer modeling, the two combination oncolytic virus virtual clinical trial that Adrian talked about on the very first day uses exactly this type of cell cycle model as the driver for most of the tumor dynamics. So it does actually occur in cancer, and that's a really nice example of how we can include immune predation, apoptosis, and also some sort of treatment into that gamma term that I mentioned on the previous slide. Okay, but I also mentioned this idea of having like a probabilistic, what's the probability that a cell is actually going to complete the cell cycle at a given time and a given age? What does that mean? Well, if we think back to the data I showed at the very beginning, it's essentially showing us, a cumulative density function for what percentage of cells will have completed the cell cycle for a given cellular age. And that's what's shown on the left-hand side here. And if we take that idea, we can actually get a nice expression for what h of t comma a needs to be. And h of t comma a is the proportion of cells that are completing the cell cycle by mitosis. And we can think about this as being the limit as delta A goes to 0 of the probability of a cell successfully completing the cell cycle inside a small window of A and then A plus delta A. And this is, of course, given the fact that the cell hasn't completed the cell cycle up to this point. So what is this telling us? Well, it's essentially saying, what's the probability that a cell that hasn't divided up until now is going to divide in the next delta A width window? And then we take the limit as that window goes to 0, and we end up with exactly the hazard rate. So if you've taken, kind of first or second year statistics, this expression is probably very familiar to you, and it's exactly giving us the rate at which cells are going to be successfully completing the cell cycle, or the hazard rate at which they're going to leave the actively dividing population. And it gives us this nice expression here where K of A is exactly the probability density function that we would integrate to get this cumulative density function on the left-hand side here. So this gives us a nice connection between our structured partial differential equation model and then the data that we're trying to recapture. And the data we're trying to recapture here is kind of this heterogeneity in intermitotic times or in doubling times. So this is nice because, well, it gives us a partial differential equation that we can throw either all the treatment terms we want to in that gamma function, or the gamma term I had on the previous slide, and then we can include the population, the interpatient heterogeneity inside this kind of hazard rate term that I'm representing as H. Okay, that's nice. We have a biological way to, or biologically motivated way to include heterogeneity in the population, but is it computationally efficient? And you might be looking at this saying, well, okay, great, Tyler, you have some sort of coupled system of an ordinary differential equation with a hyperbolic partial differential equation that I have to solve. And that's, that means I have to write a solver. This isn't particularly computationally efficient. And I don't want to do this. So this is where we get to do a little bit of math, which is kind of enjoyable. We notice that this is a first order partial differential equation, which means that we can actually solve it along the characteristic lines. And if we partially solve this partial differential equation along the characteristic lines, we end up with this nice expression for the density of actively dividing cells with a given age, where age represents how long that cell's been in the cell cycle, at a given time t. And this solution has I think, a very nice biological interpretation. We begin by taking the number of cells that were born at time t minus a in the past. Then we calculate the probability that a cell has actually survived from entering into the cell cycle at time t minus a up until time t, given that we might have immune predation, we might have random cell death, we might have some sort of treatment. And then we multiply this by the probability that this cell not only hasn't died, but also hasn't successfully completed the cell cycle, which gives us this term here. And again, if you're familiar with the statistical literature, this is the survival fraction that comes out exactly from the hazard rate that we defined on the previous slide. And this is great because once we have this nice expression for the number of cells with a given age density and time density, we can calculate how many cells are going to be completing the cell cycle at any given time. And it's exactly the product of the density of cells at time and age multiplied by the rate at which cells with this age at this time are going to be finishing the cell cycle. That's a lot to think about, but essentially what it comes down to is a cell with a given age has a probability of finishing the cell cycle. We're going to multiply these two things together and then add them all up over all possible ages. And it gives us this nice integral term here. And I kind of like this. I mean, well, I quite like this. I think this, we've done a little bit of math to go from a hard coupled ODE PDE system. We've partially solved the partial differential equation, and it gives us a fairly nice expression for the rate at which cells are going to be successfully completing the cell cycle, which is what we're really, really interested in. And because we've partially solved the partial differential equation, we end up with just a single equation for the total number of resting cells, this x of t or G0 cells that I mentioned at the very beginning. But this is no longer an ordinary differential equation because the time derivative of x at time t just depends not only on the concentration of cells at time t, but also the concentration of G0 cells at all times in the past. And the reason that we're having this dependence on the past is exactly because we think that the active portion of the cell cycle takes some non-zero amount of time. So this is what's called a delay differential equation. It's called the delay differential equation because the right-hand side of the differential equation depends on the solution with some sort of delay, or it depends on this delay term here. And so far, I've said that we should be able to estimate the probability density function K given our experimental data. That would be really, really nice in theory, but in practice, we actually normally only get kind of summary statistics. So we might get the mean of the duration of the cell cycle. That was that 33.7 hours in the example I showed at the very beginning. And then if we're lucky, we might get the variance, which is that 6.7 hours. Is 33 plus or minus 6.7. So that's nice. We get, two nice summary statistics that give us an idea of what the average time a cell is going to take to complete the cell cycle, and then how, our population might be distributed about that average. And what this means is we can use this data that we get from experimentalists to, have a good idea for what our probability density function K should be. The easiest example is to use just a Dirac distribution, which is where we would assume that every cell in the population is going to divide after precisely the mean time. This is not so nice because we tried to include this delay differential equation to have heterogeneity, and we've somehow collapsed all that heterogeneity down by assuming that every cell divides after the same amount of time. So let's not talk too much about the Dirac distribution. But the other common way that these delay differential equations are used in cancer modeling, and I say common because I've done it, we did it in that combination oncolytic virus therapy paper, it happens fairly common, is to use an Erlang distribution. And an Erlang distribution is a very specific type of gamma distribution that corresponds to essentially the concatenation of exponential weighting processes. And it comes from essentially queuing theory, so trying to decide how long you're going to wait in a line at the supermarket given how many people are in front of you. But an Erlang distribution has a very, very specific constraint, and it's a constraint that says that one of the two parameters defining the distribution must be an integer. So we must have this parameter J be an integer. But we also know that for these Erlang distributions or gamma distributions in general, the mean and the variance are sufficient statistics, which means that if I know the mean and the variance, I have enough information to say everything I want about the distribution. So we somehow imposed an imposition on the mean and the variance to make sure that our parameter J is an integer value. But the reason that we do this in practice is because we can replace the delay differential equation term or the delay term in the DDE by the solution of a compartmental system of ordinary differential equations. So we can write this integral term here as exactly the solution of this compartmental system. And that's nice because then instead of solving this delay differential equation, we can just simulate the corresponding system of ordinary differential equations. And this is computationally efficient and mathematically identical. So where are we? Well, we wanted to have a biologically motivated way of including intra-patient heterogeneity. We can do this by having a partially structured or a structured partial differential equation. And then we want this to be computationally efficient. And this is computationally efficient because we can use the standard ordinary differential equation solvers if we're using an Erlang distribution. And that's a big if that I've bolded and stressed because it actually imposes a condition on our biological data. It says that the biological data that we're getting, representing how long cells take to complete the cell cycle, has to satisfy the mean squared divided by the variance is an integer. And that's asking a lot because in reality, we should have this ratio be any positive real number. But we don't do this in practice because we can only use the ordinary different representation when J is an integer. Our statistical inference software and our virtual clinical trials are really set up to solve ODE models. And then what makes it even more complicated is there's actually no consistent numerical methods when J is non-integer. So we can't solve this equation consistently if J is non-integer. And because we can't do that, the J non-integer case isn't actually in most software packages. So we have to assume that this imposition is close enough. So this leads to a natural question of, well, why not make a numerical method? And the main problem in doing this is how on earth can we properly evaluate this delay term? And I'm going to skip a lot of the details because this is not a math talk, but essentially we're going to map this semi-infinite integral into a finite area of integration using a nice change of variables. I have conditions on how nice this must be, what they must be. I'm not going to talk about it. And then how do we do this in practice? Well, we're going to use a fourth order functionally continuous RK method. What does that mean? It's just essentially the Runge-Kutta methods that we're all familiar with in MATLAB. And then we're going to use a fifth order Simpson's rule for this mapped or this changed delay term. And why am I showing you this? Well, because we can implement this mathematical model and we can actually compare the solutions of our numerical method to known solutions of these delay differential equations. And we can look at just essentially the L infinity error between our known solution and our numerical solution. We can put this on the log scale. And the important thing here is just the slope of the line, because the slope of the line gives us the accuracy of our numerical method. And here I've shown, I think, 12 test cases. We've done this for 35 different test cases, and the slope of the error is always 4, which means we have a nice 4th order consistent numerical method, which is exactly what we wanted. But if you've ever had to read my code, you know that this is probably not very computationally efficient, and it isn't. So then maybe the question is, can we somehow have the best of both worlds? Can we somehow match both of the sufficient statistics and get away from this this assumption that the parameter j must be an integer, but then also use the established framework for ordinary differential equations? And that's the question I want to show here. And we essentially, the answer is, I'll save the suspense. The answer is yes. And instead of using an ordinary differential equation like we did in the linear chain trick, which has the same rate between all compartments, we're just going to allow this rate to vary. And what we can show analytically is that if we are given any mean and standard deviation from a delayed process, we can choose 3 rates such that this ordinary differential equation here will match both the delayed time or the mean delayed time and the standard deviation time of the delayed process that we're trying to model. So this is essentially answering yes. We can get away from this assumption that j must be an integer, and we can still use the ODE suite that is so computationally efficient. So we essentially have 3 choices when we're thinking about trying to model these intermitotic times or these kind of delay differential equations. The first is, we can use the linear chain technique and accept the imposition that we're putting on the data, that the ratio of tau squared and sigma squared is some integer. We can use this numerical method, which is accurate, but it's computationally inefficient. It's quite slow because I coded it. And then third, we can use this improved approximation that allows for rates to vary. And this comes at the cost of having to calculate 2 extra rates. So I'm going to show a comparison of these 3 different ways of thinking about analyzing or simulating delay differential equations. I'm going to use a very, very simple test problem. So it's essentially just a linear distributed delay differential equation, or a linear delay differential equation with a gamma distribution where j is non-integer. So this is not an Erlang differential equation, but rather a true gamma distribution. And then alpha and beta are just 2 positive parameters. And this is a very, very simple problem. So I can solve it. And on the left-hand side, actually in both figures, the solution we get from using the linear chain trick, which is kind of accepting this imposition that j must be an integer, is shown in purple. The true solution of the delay differential equation that I solved using my nice numerical solver is in solid blue. And then this improved approximation that comes at the cost of calculating 2 extra rates is shown in dashed orange. And I picked these examples on purpose because it really illustrates the fact that, first of all, sometimes this linear chain approximation that we assume that j is an integer is good enough can give us vastly different simulation results. And what this is showing us is that essentially sometimes our solution of the true differential equation and then the approximating differential equation can have vastly different stability properties. And that gives me a little bit of a warning that I need to be really careful about what approximations I'm using and how I'm doing that. So what should you take home from this? Well, the first idea is that, 1, we can use partial differential equations and partially solve them to get us delay differential equations that allow us to have some intra-patient or in-person heterogeneity. And then the second is that we can use existing numerical techniques or new numerical techniques to then solve these delay differential equations in a way that is computationally efficient and allows for the inclusion of this interpatient or intra-patient in-person heterogeneity in a virtual clinical trial. I've been very fortunate to work with a ton of really interesting and fun people on this. So Peter Gilch was an undergraduate student who did a lot of the numerical method. Christian Van Dorp is a postdoc at Lano as well. And him and I bounced ideas about this all over the place. And then Tony Humphries and Morgan Craig were really nice collaborators during my PhD. So there's a bunch of references, and I'm really happy to take questions. And
Thank you so much, Tyler. I think it's really great for us to have an actual really kind of mathematically focused talk, and also just to see how useful DEs can be. And I think it's important even for me sometimes to remember that I don't need to be afraid of trying to use something that might be computationally difficult to solve. So I think it's very exciting. So thank you so much for sharing all of that with us. So we have one question in the chat, which says, can you please share the Stato and all 2016 reference?
Yes, it's here. So it's 2016 in SciRep, scientific reports. I can put that in.
It might be here now. And was this human data?
I believe so. I'm fairly certain it was from the HeLa cell line, so Henrietta Lack cell line, but I will have to double check. It was done in vitro to answer that question, I guess.
Okay. Well, yeah, if you're happy to share that reference or the name, then Rajat can have a look and find out. And so Lizette just asked a question in the chat as well. She said she really likes the numerics and mathematics. From a user's perspective, can you say something about how DDE 23 in MATLAB may or may not be well suited to solving your systems of equations?
Yeah, so DDE 23 in MATLAB is a nice code. Where am I? Here. So DDE 23 is a nice differential equation that's been implemented in MATLAB. It's a third order RK solver, so it's one order less, but 3 is often good enough. But the DDE 23 is actually specifically set up to solve these discrete delay differential equations, where we assume that essentially all cells would be dividing after the same amount of time in the cell cycle. So it's a very good solver if this is the type of modeling you want to do. It unfortunately does not work very well if we're thinking about these distributed delay differential equations. But the theory underlying these discrete delay differential equation, which is called the functionally or continuous Numerical method, is the same. It's just the implementation is a little bit different.
Right, gotcha. Okay, cool. Julio just asked a question. Could adaptive time step solvers be suitable for solving DDEs like ODE 45 in MATLAB?
Yes, yeah, so I didn't show those results here, but essentially the adaptive time step sizers, it basically comes down to like a predictor corrector algorithm where the solver will choose a step size and then try to correct that step size, and if it's good enough, you'll accept it, and if not, you'll choose a smaller step size. And that's how these adaptive time stepping methods work. And there's nothing specific about that idea to ODEs, so they're actually in theory simple to implement, in practice a little bit more difficult, but the idea is exactly the same and it can be done. And that is what DDE 23 does.
Great. Awesome. Well, thank you so much for all that. I think that was really interesting. We all really appreciate it. And I'll let Morgan introduce the next speaker. And thank you, Tyler, again for your great talk.
Thanks.